Okay, so this is chapter 17, and it's going to cover controlled cardiac function, cardiovascular function. It's basically it's stuff that you need to know. Hopefully, some of it though you'll recognize from physiology, from your regular physiology class. But there's still things that we need to know in here, and so we'll probably go into a little bit more detail than you did there. Okay, so first of all, the cardiovascular system, as we probably know, consists of the heart, and it has arterial vessels, so these are our arteries, and venous vessels, which are our veins. So the arteries go away from the heart, so that's something that's moving out, and the venous are going back toward the heart, so they're returning blood to the heart. The functions, we usually, when we think of cardiovascular and we think of what the blood is doing and what the blood is carrying, we usually think of oxygen, but it's so much more than that. It's oxygen, glucose, fats, cholesterol, amino acids, signaling molecules. So we've talked a lot about hormones and cytokines. All of these things also move through the blood. And then, of course, we have white blood cells, clotting factors, remember, that were made in the liver. All of those things also moving through the cardiovascular system, through the vessels. And they also remove waste. So every metabolism, when a cell is functioning, it's going to there's going to be some kind of waste products in addition to carbon dioxide, which we have listed here. You're also going to have breakdown products and amino acids and things like that, just like acetates, certain molecules that are just sort of thrown off from these chemical reactions. Uh, so um, so metabolic waste, and then a lot of these things are broken down and then excreted by the liver. Some of them are filtered by the kidneys and then excreted as urine. So we have the renal system, which is filtering the blood, that we'll get to talk about at a later time. Okay, so structure of the heart. A couple of things. The Because we'll hear about things like endocarditis, which is the endocardium, which is the inner layer of the heart. This one is really important, the myocardium. Okay, so the myocardium is what we refer to when we're talking about the muscles, the individual, those striated muscle cells that make up the heart, most of the heart. And we call that the myocardium, myo referring to muscle, and of course cardium is heart. And then there's the pericardium. The pericardium is a sac that is sealed around the heart. Okay, so the heart is actually beating inside this sac. So it's fibrous on the outside and then it's serous on the inside. And so the heart kind of always has this nice little lubricated fluid that it's beating in. But sometimes issues can come up with the pericardium as well. Okay, so you can see from this picture, you can see the valves, and I'm going to, and you can kind of imagine if we cut right through, that these valves are actually about in the same plane. So we can kind of see that a little better here, and you're going to have to know all of these valves, and you're going to have to know their alternate names. Um, so if we start with the valves that are between, so we have the atria and we have the ventricles. The valves that are between the atria and the ventricles are called atrioventricular valves. Okay, so blood is going to flow from the atria into the ventricles. That's what it does, and it flows through the atrioventricular valves. So there's a left AV valve and a right AV valve. The left AV valve is usually just called the mitral valve. Okay, so that's going to be on the left side, which is going to be part of systemic circulation. That's where the left ventricle squeezes out to the system, so it's going to go not to the lungs. So the mitral valve is on the left side, and we also call that the bicuspid. Now the right side is usually referred to as the tricuspid, or the right atrioventricular valve is usually referred to as the tricuspid. Okay, so that's on the right. So mitral, tricuspid. The mitral is also called the bicuspid, and then they get their respective left AV or right AV. Okay, so now leaving the ventricles and going out either to the aorta or to the pulmonary valve are the semilunar valves. Okay, semilunar because they look like part moon. Okay, so that's why they were called semilunar valves. And so that's flow from the ventricles And on the left side, going to the body, we call that the aortic semilunar valve, okay, because the first place it goes is into the aorta. Okay. On the right side, it's going to the lungs, so we call that pulmonary semilunar valve because it's going to the pulmonary artery. Now, pulmonary always refers to lungs, okay? So just remember that, put it in a special place in your brain, and try not to forget that, because anytime you're talking about pulmonary, you're talking about lungs, okay? So a couple of other things. The papillary muscles, okay, so you can see these little muscles down here. These are papillary muscles. And what they do is, if you think about how much pressure, especially on the left side, where that left ventricle is squeezing out to the entire body, it's creating a lot of pressure. Now, these valves, they have to, these, they, they could what's called prolapse. Okay, so they could, when the ventricle squeezed, these AV valves. So if we have a atrium here, and then the ventricle here. So it's possible when the ventricle squeezes that these things could just blow back this way and go, and the blood could flow right back into the atria from where it just came, and we don't want that to happen. We want it to go out through the aorta. So to prevent that prolapse, or prevent that valve from going the wrong direction and just allowing blood to go back, we have these papillary muscles that are uh, attached by these tendons, and they will hold that. So when the ventricle squeezes, these guys, which are kind of continuous with the ventricles, will squeeze too, and they will hold that valve to keep it from prolapsing, or to prevent blood from being regurgitated back up into the atria. Okay, so those guys are important, and if they fail, then you're going to have that regurgitation. You're going to have blood flowing back into the atria, and then that's not an effective heart. That's one of these things that could lead to failure of the heart. Okay, so this is the blood flow through the heart, and these are all the things that you're going to have to know. You're going to have to know where blood is flowing, when it's oxygenated, when it's deoxygenated. It's really, aside from learning the terms, it's pretty straightforward. I mean, if you just pick a place to start, in this case we're starting at the right atrium, it goes from the right atrium to the right ventricle, because the atria always go to the ventricles. The right side is going to go to the lungs, okay? Well, in the lungs it picks up oxygen. So you can see here that it turns red because now it has oxygen. Well, it's not going to go back to the right side. It's going to go to the left side now. So the oxygenated blood goes to the left side, again through the atrium. Atrium always squeezes to the ventricle, and then the left ventricle goes out to the body. So once you understand the flow of blood, then you kind of have to go back and you have to memorize these valves we just talked about. Like on the right side, it's the tricuspid or the right AV. On the left side, it's the mitral valve or the bicuspid or the left AV. And then you also need to remember your semilunar valves. So if it's leaving the ventricles, it's got to go through one of the semilunar valves, either the aortic for the left side or the pulmonary on the right side. Okay? So just make sure that you understand this. Okay? That you understand exactly how blood's flowing and those details that we just kind of talked about. Okay, blood vessels. So circulation relies on blood moving through vessels, obviously, and we actually have two systems. I've kind of alluded to that, but I haven't officially said it. But we have two systems. We have blood that's circulating through the body, and then we have this other system where blood circulates through the lungs, and so we call those the systemic system or the pulmonary system. Now, usually when we think of arteries, we think of oxygenated, but that's because we tend to focus most on this systemic. So if blood is flowing away from the heart, we think, oh, it's going to have oxygen. Okay? And so arteries have oxygenated blood. Well, arteries are also associated with blood that's going to the lungs to get oxygen. So those are arteries that are deoxygenated. So the systemic, it's really the same. The systemic, it goes through the aorta first, and then that's the main vessel that's coming out of the heart. And then arteries like the brachial artery, the femoral artery, or something like that. And then those get smaller and they turn into arterioles, which are small arteries. And then those will turn into capillaries, which are just one cell layer thick. This is where exchange takes place, like gas exchange, nutrient exchange. That's where that takes place in the capillaries. So we can see here it goes from being oxygenated to deoxygenated because it gives up its oxygen. And then those, after it goes through the capillaries, that will expand and turn into venules, and then veins, and then systemic, it goes to the vena cava. So pulmonary is the same thing, except that as it leaves the heart, it goes through the pulmonary artery, and then again, arteries, arterioles, and in this case, in pulmonary circulation, it becomes oxygenated. It goes from being deoxygenated to now oxygenated, and that happens in the capillaries. Again, that gas exchange. Okay, and then venules, veins, and then the pulmonary vein. The pulmonary vein is that that vein that's going back to the left side, so it's back to the left atrium. Okay, so blood moves fastest through the arteries and veins. Okay, and that's what this graph is showing. Okay, so it's showing the velocity, that's down here. It's showing the velocity, it's showing that it's faster in the arteries and it's in the veins, so it's absolutely faster in the arteries, which makes sense because the heart is pumping that blood. So it's fastest there, but then it slows way down in the capillaries. Okay, notice that we're talking about velocity, not pressure. Pressure continually gets lower and lower as it moves through the system, but as far as velocity, it gets very slow in the capillaries because it has to, because that's where exchange is taking place. Okay, so we want to give plenty of opportunity for that oxygen to leave and the carbon dioxide to come back, or the nutrients or whatever. Okay, so that's where exchange takes place in the capillaries, and it moves slowest through the capillaries because they have a large cross-sectional area. Okay, so if you were to cut all of the vessels, just cross-cut them all and then measure from one end to the other and add all of that together, what you would see is you would see that capillaries would be huge. Even though each individual capillary is tiny, when you put them all together, that cross-sectional area is going to be enormous. So it's like when a river kind of flows out into like a really wide area, and the river may be running very quickly, so you may have rapids, and then it hits that wide area and it just slows way down. Okay, and it just barely moves along. And then when it constricts again, then the velocity will again increase and it'll be fast again. Okay, so that's what's happening from arteries to capillaries and then back to veins. Okay. Now, laminar and turbulent flow. These are, they sound like fancy names, but there's really a couple of main concepts here. Laminar, first of all, means layered. And so you can see with laminar flow that everything is going along nicely in this very ordered way. It's just flowing. There's not a lot of what we would call turbulence, because there aren't any interruptions. Okay, so there's going to be some resistance from the outer area here along the vessel, because it's going to be quite literally scraping along the vessel. Of course, they're molecules, so they're kind of bouncing along, but that's an interruption. And so what that means is that flow, and that's what this picture is trying to show you, flow is faster in the middle than it is along the walls of a vessel. Okay, and that's because there's some resistance over here from the actual blood, in this case, hitting the vessel wall. Okay, so that means this in the middle is flowing along nicely. Turbulent flow is different. That's interrupted, and so when you have like a constriction, which we can see here, if you have a constriction, then that blood that was flowing nicely is all of a sudden quickly redirected, and it causes it to swirl. Okay, so basically it's causing it to swirl, and then instead of blood flowing nicely in this nice layered or laminar flow, now it's kind of spinning around, it's bouncing off the wall, and so that's going to be a lot more resistance. It's just a lot more chaotic. Okay, now we can hear that, and that's why this is partially responsible for heart sounds. So when like when a valve closes or something like that, we can hear that sound when the blood starts to kind of swirl around and starts bouncing off of the vessel wall. We can hear that vibration, and that's what you can pick that up with a stethoscope. Okay, sounds are due to turbulence. Okay, which is not normal in arteries. So we shouldn't hear a lot of turbulent flow in arteries, and it's probably due to some kind of a constriction. So if there is a constriction, then you're going to hear turbulent flow. Okay. Now, we also learned that turbulent flow can increase the probability of thrombosis. So these platelets can start to aggregate at areas where there's a lot of turbulent flow or a constriction or something like that, which is a problem with things like atherosclerosis, where you have some kind of a constriction and then it interrupts the blood flow, and then those platelets will start to aggregate to form a thrombosis or a clot. Okay, so I have down here important blood contacting the side of the vessel increases resistance to flow. Okay, so more resistance because it's kind of, like I said, scraping along the side. Of course, it's blood, so it's not really scraping, but it is interacting. Okay, when the vessel has a larger diameter, more blood is allowed to flow unimpeded. Okay, so that kind of makes sense too. If you've got a vessel that's very small, you're not going to have very much room, and you're going to have comparatively a lot more blood scraping along the side of the vessel. So when you look at the proportion, you're saying, wow, these small vessels, yeah, there's a lot of scraping and just a little bit of blood in the middle. But when that blood vessel is huge, yes, you still have some resistance along the vessel wall, but you've got a whole lot more of that middle area. And so you have a whole lot better flow. Okay? And this is one of the reasons that increasing the diameter of a blood vessel really increases the amount of flow that it's capable of and will decrease pressure, okay, when we make those bigger. Okay, so heart sounds result from turbulence. Normally, if a valve is open and it opens nicely, there's going to be very little turbulence, so it's going to be quiet. But when it is stenotic, and that's a term you need to remember, stenosis or stenotic means that it is a narrowed valve, which means that there is going to be an interruption, and you can see in this image that you have a lot of turbulence. Okay, so you have this turbulent flow, and that's going to contribute to the sound. So if you have like a mitral valve or something like that isn't working properly, it's stenotic, it's not opening all the way, then there's going to be flow. It could be prolapse and blood could squirt out, which is going to be turbulent flow, and you're going to just hear that as an extra little sound, a lot of times a little squish after it's supposed to stop, and that's a murmur. Okay, so we can hear a heart murmur, which indicates that there might be some kind of a valve problem or something that's causing that turbulent flow. Okay, so a stenotic valve is kind of twisty there. An insufficient valve means that it's really not closing all the way, and so it's supposed to stop the blood from flowing. And that's what I was just mentioning. If it doesn't close all the way, then that blood can squirt back out, and that's also going to be heard as a murmur. Okay, so pressure, resistance, and flow. So we already, I think most of us already, because we've talked about it in other classes, but I think we kind of already understand this concept, that if a blood vessel is larger, the pressure is going to be decreased, because we talk about vasodilation, and if we have vasodilation, then that's going to lead to a decrease in blood pressure overall. So we're going to spend the next couple of slides just kind of talking about why that's the case. Okay, so fluid flow through a vessel depends on the pressure difference between the ends. Okay, so where are you going to have the maximum amount of pressure? Let's say we're talking about systemic blood flow. You're going to have it at the aorta, right at the ventricle. The ventricle is going to squeeze. That's where the most of your pressure is. Well, since blood keeps flowing, obviously the pressure is going to be higher behind it. It's going to flow from high pressure to lower pressure. And so that means that's why we can confidently say that blood flow is always going to be highest in arteries because it's just going to decrease as it flows through the system, and it's going to be at its lowest when it gets back to the heart after it's traveled all around the body in the case of systemic. Okay. All right, so pressure difference between the ends of a vessel. The heart creates the pressure, yes. Moves from a higher pressure to a lower pressure, yes it does. And that pressure is what pushes the fluid through. Pressure also, this maintenance of pressure and this volume is also going to keep the vessels from collapsing. Okay, if they were not under pressure, if you were leaking out blood so quickly that you weren't able to maintain any pressure, then the blood vessels could collapse. Okay, so the vessel's resistance to flow, small vessels, like I just mentioned, have more resistance. Okay, and larger vessels have less. That should say less resistance. Okay, less. So smaller vessels have more resistance, larger vessels, oh wait, oops, no, my mistake. Small vessels have more resistance, larger have less, but longer. This is longer, not larger. Longer vessels have more resistance, and that's just because you have more opportunities for that resistance. If it's longer, you're going to have more scraping along the side. I know I say that, and it's not really what happens, but it kind of gets the point across that it's interacting with the side. And so the longer that goes on, the more your pressure is going to decrease because that's just kind of an increase in resistance. The more viscous or thicker, so when we talk about viscosity, something that's very viscous is very thick. So that means if we have a whole lot more red blood cells in our vessels, then it's going to be thicker. So if these are red blood cells, that's going to be one viscosity, and that's going to have a certain resistance. But then if we try to push a whole bunch more red blood cells in there, or white blood cells, it doesn't matter, any kind of blood cells, or anything really that's going to make it thicker, but usually it's blood cells that do it, then that's going to increase resistance. You increase resistance, you increase pressure. Okay. So, um, so increasing diameter decreases resistance. That's Poussin's law. And this is the important thing to understand, is that doubling the radius does not just cut the resistance in half. It actually reduces it by a factor of 4. Okay, so resistance is proportional to 1 over the radius to the 4th power. Okay, so since we're not going to be calculating this, we need to understand what that means. So that means if I have 1 over 1, that's my size here, whatever. But if I cut that in half, it's actually going to be a 16-fold difference. Okay, so if I go from a radius of 1, that's what this is showing. If I go from a radius of 1 to a radius of 2, which means I doubled the radius or the diameter, but the radius is the halfway point. The diameter is all the way across, so the calculation uses radius, just does. But if I double the radius, then that is going to actually increase my flow by 16 times. So I'm going to have a 16-fold increase in flow just by doubling it. And if I triple it, then it's much, much higher. Okay, so we kind of bring all this together here in this calculation, and it says flow times resistance equals a change in pressure. Okay, so this is where we get it. This is why we say that if we vasodilate, our pressure drops, and it's because our resistance drops. Okay, so if we have less resistance, then this number is going to be lower. Flow times resistance, yeah, we decrease that resistance, our change in pressure, we're going to have a lower pressure. Okay, so if resistance increases, pressure increases, as long as your blood flow stays the same based on that calculation. Okay, so again, same thing. Relaxed vessels decrease resistance. Vasoconstricted vessels, so vasodilated, so relaxed are vasodilated, they decrease resistance. Vasoconstricted increase resistance. And this is the biggest contributor to minute control of blood pressure in the cardiovascular system. Okay, so it is this resistance, or this vessel diameter. And plaque buildup inside blood vessels contributes highly to this, which is going to, it's going to decrease the radius, therefore it's going to um, increase the resistance, and you're going to have more. So it's going to decrease the radius, increase its resistance, and therefore your blood pressure is going to be higher. Like I said, we've kind of been using this, and we've been talking about this all along, but now we kind of are seeing how this is actually calculated, and that's actually a 16-fold change based on radius. Okay, so what does it all mean? Small changes in diameter equal big changes in flow, and assuming the output of the heart does not change, this has a dramatic effect on blood pressure. Okay, and this again shows the 1, 2, and then when it doubles in size, 16 is the new flow. Okay, so the heart produces a pressure gradient. I've already mentioned this. So blood has to flow from a high pressure to a low pressure. That's just the law of the world. You don't see something going from a low pressure to a high pressure. You can't do it. So things like air is the same way. If it's really pressurized, air is going to flow out towards lower pressure. So therefore, since the highest pressure happens in the heart, then it just decreases as it goes along. Okay, so that means, this question here, if you think about that and you know where your vessels are, this is pretty easy. Where's pressure greatest, arteries or capillaries? Well, it's going to be greater in the arteries, okay, because they come first. They're closest to the heart. What about capillaries or veins? This likes to throw people because we know that blood flow velocity is lower in capillaries, and so we might want to say, oh, well, it must be lower pressure, but it's not. Overall, yeah, yeah, the pressure is going to be low where the velocity is low, but there are so many of them. But when you sort of add up all of the pressure of all of the capillaries, it's overall going to be greater pressure. It's just spread out more. Okay, so the pressure decreases as we move through. So that means that veins are going to have lower pressure than capillaries. And think about it, it has to be that way. If capillaries had lower pressure than veins, blood would be flowing from veins into capillaries, okay? And that doesn't happen. All right, so electrical stuff, we need to talk about that. So here's a quick overview of action potentials. And action potentials, a couple of things. First of all, what is an action potential? Well, moving down a muscle, so this could be a muscle membrane or it could be a neuron, but whatever, you have depolarization, repolarization. Moving all the way down. It depolarizes or repolarizes, wherever it is on the membrane. Okay? And that signal will then move in a certain direction. Okay? So it can move that way through cardiac muscle, which is how it's going to spread, and then, or it can move that way in neurons, which is how a neuron communicates. Okay? So if we look at what happens in one of these depolarization, repolarization, it's actually fairly simple if we just sort of accept a few things. Like, first of all, the membrane potential is always sitting at -70, and that's usually because, potassium can move in or it can move out if it wants to, and the sodium-potassium pump is kind of keeping the sodium and potassium levels where they're supposed to be, but it keeps it at about -70. When a depolarization starts, that means some positively charged ion was let into the cell. So if this is a myocardial cell, then it's sitting here at -70 to begin with, and then suddenly sodium, it's usually sodium that depolarizes, starts to move in, and this stops being -70. Now it starts to increase. So it goes from -70, it starts to go up a little bit, until it reaches a threshold. And then when it reaches a threshold, all of a sudden voltage-gated sodium channels open, and this is the important thing, sodium rushes into the cell and it causes it to depolarize. So that's what happens here. Sodium is rushing in, I'll put rushes in, that way you know that that's what I'm talking about. So sodium rushes in, and that's depolarization. Well, then, at the same time, potassium channels start to open. Now, potassium was already at a higher concentration on the inside. I guess I should have mentioned that. If you forgot, remember, sodium is at a high concentration outside, potassium is at a high concentration inside. So now, sodium rushes in, and since the voltage became clear up here, it went all the way up to plus 30. Well, at that point, sodium channels open. They were starting to open before, but the sodium channels open and allow sodium to move out. Okay? So since sodium is moving out and taking its positive charge with it, then suddenly the membrane starts to deep repolarize. So it goes back down to minus 70, and it actually overshoots because sodium really wants to get out more than we probably realize. And so when it's just allowed to come out, then it goes all the way down, hyperpolarizes, or we call that an overshoot, and then the sodium potassium pump comes back in, fixes everything, and it's ready to go again. So it's ready right away in a normal neuron. It's right away ready to be depolarized again. Now we're going to see that in a cardiac neuron or a cardiac muscle cell, the depolarization, the repolarization starts, but then calcium starts to move in, and it keeps it depolarized for a longer period of time. Okay. All right. So I'm not going to read through all this. You do have to know what an action potential is. You do know how they have to work, but that's a lot to just read. So you can read through all that. Okay, so how does the heart beat rhythmically? We care about this. We care about how the heart beats rhythmically because our brain isn't sitting there for every beat of the heart telling it, okay, beat again, and beat again, and beat again. It doesn't do that. The heart takes care of it all on its own. And so I sort of mentioned the experiment where we actually removed the heart from a frog, and it kept beating in this Ringer's solution that had all of the ions where they were supposed to be, and that it just kept beating because the heart itself has what are called pacemaker cells, or autorhythmic. Autorhythmic means they set their own rhythm. Now, the brain will control that. The endocrine system can control that. So, your fight or flight, your sympathetic nervous system can turn it up, your parasympathetic turns it down. But overall, the actual beating is done and controlled by the heart itself, okay, with these pacemaker or autorhythmic cells. And this is something that you need to remember. People get this mixed up all the time. When we're talking about autorhythmic cells, we're talking about a tiny little part of the heart. Okay, so it's this tiny little area, and there are a few of them. And like the Purkinje fibers, they're fibers. So in this case, we're looking at what's probably the SA node, and it's just this little button of cells that depolarize by themselves. Then they will spread that depolarization to the myocardia. So when you're looking at this, okay, so pay attention here. These are the depolarization repolarizations of the autorhythmic cell, but when we're talking about depolarization of myocardia, we're talking about the muscle cells. So this is what an EKG looks at. Okay, an EKG looks at the myocardia. It does not look at the pacemaker cells. It could probably detect them, and I'm sure some EKGs are powerful enough that they can see the depolarizations that happen in the autorhythmic cells. But normally when we're looking at an EKG, we're looking at the massive, just tons, okay, tons, I'm exaggerating, but there are a lot of myocardial cells compared to these autorhythmic cells. So the autorhythmic cells just depolarize and repolarize. That's their whole job, okay? And then they spread that signal through these things called gap junctions, which are they're junctions holding the myocardia together, so that they're holding these individual cells together, but they leak. They have these gaps in them, so they call them gap junctions. All right, so a little bit about autorhythmic cells. So the heart is responsible for its own contractions. The autorhythmic cells are small groups of cells that depolarize spontaneously by leaking calcium. They also leak sodium. So there are a couple of channels here. I put them up here, so just if you're visual, you can kind of see what's going on. And so you have these cells that are constantly leaking in sodium first and then calcium, okay? So they start out leaking sodium and then they leak calcium. And that's going to allow them to reach this threshold. Okay, so if we move down to this graph, that leaking of the calcium at first lets them get to this threshold level, okay, and then they will leak full on from L-type long-term calcium channels. That's what causes the depolarization. Now remember, when we were talking about muscle cells, we were talking about sodium as causing the depolarization. Okay, remember, we're still talking about these smaller groups of autorhythmic cells or pacemaker cells. In the pacemaker cells, this is what confuses everybody. They get the pacemaker cells mixed up with the heart muscle cells or the myocardium. In pacemaker cells, it's calcium. So the actual depolarization of autorhythmic cells occurs because calcium rushes in. Okay, and it's these L-type, and guess what? We have these drugs called calcium channel blockers that block these L-type calcium channels. And so it lets the calcium in a little slower. So if we were to look again at this graph, we would see that while it was trying to depolarize, it's going to happen a little more slowly. And so that's going to slow the heart rate. Okay? So it's not, so those autorhythmic cells are going to depolarize more slowly, slows heart rate, and that's going to contribute to reducing cardiac output or lowering blood pressure. Okay, so again, spreads a signal to the myocardium, which is the heart muscle, through gap junctions. All myocardial cells must depolarize at the same time. So that's something else to remember, is that I know this is just a small section of the heart, but once these depolarize, it spreads all over. And there are some pathways that help it spread more quickly that we'll look at. But all of the atria have to depolarize at the same time. We know this because both the left and the right both squeeze to the ventricles at the same time, and then both ventricles will squeeze at the same time. So the atria have to contract at the same time, and then there's a pause, and then the ventricles all contract at the same time. Okay, and so it does this through, and since I'm talking about it, I'll go down here and look at this little thing with the green tracts, and these are internodal pathways. So if we have depolarization at the SA node that takes place right here, then it can spread to the atria, and all of the atria then can contract at the same time because it also spreads to those gap junctions. Well, then it also moves down here to something called the AV node, okay, and then it will pause, okay, and then the ventricles will all contract at the same time. Now, when we're talking about setting the heart rate, normally we have this group of cells, this pacemaker node called the SA node or the sinoatrial node. That's the one that in most people all the time is setting your heart rate. These are the ones that are depolarizing on a regular basis from 60 to 90 beats per minute, something like that. Okay? Now, the question is, what if it fails? What if our SA node fails and doesn't work for whatever reason? Then you have another node, autorhythmic cell, or group of cells, called the AV node, atrioventricular. And so now we can look at this one. So here was our AV node right here. Okay, that's our AV node. Well, if it fails, okay, then this one, or I'm sorry, this was the SA node. SA node is the first one, so that's there. So if the SA node fails, then the AV node will kick in. It's a little slower. Okay, so the AV node is only about 40, it will spontaneously depolarize about 40 to 60 beats per minute, so it usually doesn't get a chance. Usually the SA node depolarizes, and when it depolarizes, it immediately sends a signal down to the AV node saying, nope, I got it, and the AV node will reset. Okay, but in the event that it doesn't work, that AV node can kind of step in, and it will be a slower heart rate, but it will still maintain a heart rate. Okay, so that's a good little backup plan. Okay, so the next question is, or the next point is, what if our SA node fails and our AV node fails? Okay, does that mean the ventricles, okay, can't contract? Because remember, I said the signal moves from usually the SA node, then through the AV node, and then it moves through this little area right here. Okay, and that's called the bundle of His, or the AV bundle. Okay, so it usually, when these depolarize, it moves through this area and pauses. Okay, like a 0.1 second pause or something like that, and then it continues on into the ventricles. So if our SA node fails, okay, that one fails, the SA node fails, and the AV node fails, it's not going to send a signal. Okay, I can tell you that if these both fail, it's not going to send a signal to the ventricles saying to contract. Okay, it's just not going to happen. But will the ventricles contract? And with this experiment with this little frog heart, we actually removed the atria from it. We cut right across there, and we had the ventricles isolated, and they did contract. And that's because we have this third, or some books call it the third and fourth, but it's this network here of autorhythmic cells in the ventricles, and that's the AV bundle, bundle of His, bundle branches. So these all can depolarize as well by themselves, but they do it much more slowly. Okay, so the Purkinje fibers are usually credited with having the autorhythmic capacity. So the Purkinje fibers, which sit down here, there are these little fibers that are kind of down here around. They kind of surround the ventricles all over, and they can spontaneously depolarize, but they do it very, very slowly. Okay, so only 30 to 40 beats per minute. Okay, which, if you have someone who has some kind of heart failure, or let's say they've had heart damage that's blocked the communication between the SA node, the AV node. It's blocked communication between the atria and the ventricles, so that signal isn't moving out anymore. Then you might see on an EKG that the depolarization of the atria are taking place, but then it's not being followed by the depolarization of the ventricles. Okay, so you'll see these humps that go along on a regular basis, and then every now and then, very, very slowly, you'll see a ventricular contraction, and that's because you have these autorhythmic cells down here in the ventricles, which include that pathway, the atrioventricular bundle, or the AV bundle, also called the bundle of His, and then that projects to these Purkinje fibers, and these Purkinje fibers are autorhythmic. Okay, so all of these cells are considered autorhythmic, but slow. Okay. So communication between the nodes occurs through the internodal tracts. The only path, and remember that, the only path from the atria to the ventricles occurs through the AV node and the AV bundle or the bundle branches. Okay, so that's where the communication takes place. And why is that important? Because we have partial blocks. We have something pathology. It's called a partial block, or you have blocked the track, this electrical connection between the atria and the ventricles. And like I was just showing you, that's what shows up on an EKG. So even though you've got the atria are working, your ventricles are lagging way behind. Keeps you alive, okay? So it keeps some blood flowing, but you're going to have to, that's going to have to be fixed very, very quickly. Okay, so there's another problem. We don't want a heart muscle to contract and then just stay contracted. We want it to contract and then we want it to relax. So we want contraction, relaxation. Okay? We don't want to do like we can do with a skeletal muscle and start to contract it and then send another impulse that says, hey, contract again, where it just stays contracted and contracts maximally and then stays contracted. Like if you flexed your muscle and just kept it flexed, that's fine for a skeletal muscle. We don't want the heart to do that. We want the heart to contract and then relax. Okay, so the way the body has figured out to do that is that when this first action potential comes, and remember, it's communicating all of these cells in the myocardia. If we just sort of focus on the atria for a minute, all of the cells in the atria, both atria, are all depolarizing at the same time. but they also leak to each other. So we don't want this one to depolarize and then send a signal back and all over the place, and all of the heart muscles are confused. We want a simple depolarization one time of all the cells and then for them to relax. So rather than having an action potential that looks like this and then straight down, we have an action potential that looks like this and then stays depolarized for a little while. That gives the myocardium a chance to contract and then relax before another contraction can take place. Okay, so we call that the refractory period. And the refractory period is that period of time where nothing can happen. So the example being when you flush a toilet, and then while the toilet is flushing, you jiggle the handle, you can't flush it again. Okay, so that's the refractory period. And it's longer in cardiac muscle, okay, because we don't want to have summation. We don't want that to max out. We don't want that contraction to max out. We just want it to contract, relax. Okay, and so it does that by this one very, very special thing, and that is it allows calcium into the cell. Okay, now that doesn't happen in skeletal muscle. In cardiac muscle, you have these little calcium channels. They're L type. Again, remember hearing about those. L are long lasting. That means they stay open for a while. So L-type calcium channels open here too. And so we have depolarization, which, remember, in myocardia, depolarization happens the normal way. Sodium coming in. It was in those autorhythmic cells, remember, that calcium was. Don't get those mixed up. Okay, so sodium rushes in, and then normally potassium would rush out in repolarization. But because we have these little calcium channels here that are annoying everything, it stays depolarized for a longer period of time, and then the potassium moves out and you have repolarization. Okay, so by allowing calcium into the cell, the membrane remains depolarized for a longer period of time, which creates this plateau. Okay, so it creates a plateau. So straight down like normal, and then you're ready for another one. It creates a plateau. All right, during this plateau period, another action potential can't take place. So any signal saying to beat again is just ignored. So passing around those signals isn't going to matter. Okay, everything's going to depolarize and then pause and then repolarize. Okay, we call this a longer refractory period, which we can kind of see. We'll see on another slide as well. Okay, so what's going on? A little bit easier picture maybe to understand is that you have depolarization of the myocardial membrane, and then calcium channel opens. Okay, so depolarization is already happening. Sodium is moving in, potassium moving out, whatever. But then because it's a myocardium, this depolarization is going to lead to calcium moving in, and that channel is going to stay open. So instead of repolarizing right away, it just stays depolarized, and it allows the muscle, that's what this is showing, if you remember that, the muscle contraction to take place. It allows that calcium to bind to troponin, move tropomyosin out of the way. Myosin heads walk along actin filaments. That's the way the contraction takes place, okay? And it stays depolarized the entire time because of calcium. Okay, how many times have I said that? I hope that makes sense. All right, so autorhythmic cells depolarize and spread to the myocardium. Okay, so that was out here. Remember the little autorhythmic cells spreads to the myocardium, and then depolarization of the myocardium will then allow calcium to enter. So it's depolarizing the same way. First sodium moves in, but then it allows calcium to enter. And then that calcium is going to extend that contraction. So calcium influx through these L-type calcium channels activates, it keeps it depolarized, but it also does the rest of it. It activates release of calcium from the sarcoplasmic reticulum, because remember, this is just a muscle cell, and then calcium allows that contraction to take place. And of course, muscle contractions, myosin walking along actin filaments. Okay, so calcium channel blockers are going to be involved in this too. Remember how calcium channels blockers were involved in the pacemaker cells, because the pacemaker cells depolarize with calcium. Okay, So it was affecting that and slowing pacemaker cells down. Well, the actual myocardium, it will block those L-type calcium channels too. So it's not going to let the calcium in as quickly, or actually what it does is it extends its entry for a longer period of time, which means that you're extending the refractory period for a longer period of time, which also means you're going to have a slower heart rate. Okay, so calcium channel blocker blocks it, slows the entry of calcium into the myocardium. A certain amount of calcium has to enter before it will close, so it lengthens the refractory period and slows the heart rate. Okay, and then again, remember that also works for the autorhythmic cells. Okay. All right, so again, one more picture to kind of drive home this message. A single contraction without summation is required, and so the longer refractory period allows that. So we have depolarization, or we have this depolarization that occurs, so that's just sodium rushing in, at least in myocardia. In the autorhythmic cells, it was calcium. So depolarization happens, and then you have that little lag until the calcium can start to cause the muscle contraction. But during that period of time, more calcium is let in, and it keeps the refractory period longer. So no action potential can take place again until right about here. Okay? And then you can have another action potential and another contraction. And so just a little note to see here. I have this little question down here. What is the maximal rate for this heart? Well, if the refractory period lasts, this is 250 milliseconds, or a quarter of a second, okay? Assuming 1,000 milliseconds in a second. So a quarter of a second, if that's 1/4 second, oops, and I write 1/2. So if that's a fourth of a second, that means the maximum this heart's that we're seeing here is going to be able to beat is about 4 times per second, which is about 240 beats per minute. So that means that you can actually have a maximum heart rate, and you do have a maximum heart rate. Okay, and it all depends on the length of this refractory period. Okay, so it's all kind of related. It all goes back to calcium channel blockers, and that's why we care. Okay, all right. So and um, calcium content in the blood, so your blood calcium levels can affect that as well. So yeah, it all kind of ties back together. Okay, so the EKG shows massive depolarization of the myocardia, not the pacemaker cells, the myocardia. Okay, because that's what the heart is mainly made of. That's what you're going to see the most. Okay, so the P wave is depolarization of the atria. The QRS wave is going to be depolarization of the ventricles. Okay, remember, ventricle depolarization happens afterwards. Okay, so all of those cells are going to go about at the same time. And then the T wave is repolarization of the ventricles. Okay, so those are the electrical things that the EKG can sense. Okay. So depolarization of the atria, well, if you have depolarization of the atria, you expect repolarization of the atria, and it does happen, and it happens right here, buried under the QRS complex, and you don't see it. Okay, so it's a very, very tiny signal. The repolarization signal is a little slower, and so it doesn't show up as well on an EKG. So repolarization of the atria is masked. That's what we say. It's masked by the QRS complex. QRS complex, depolarization of the ventricles. Repolarization is the T wave. And then down here, the U wave, you might see that for like repolarization or depolarization of the papillary muscles or some other parts of the ventricles that is timed a little differently. Okay, so we can also tell something about intervals. Now, if we have the P wave, which is depolarization of the atria, and then there's a long delay until the ventricles depolarize. Think about that. Just think about that for a second. Depolarization of the atria means the atria are contracting. Now they send that signal through, when the SA node depolarizes, it sends that signal down to the ventricles. What happens if we have a bit of a block and this is delayed? What if instead of being here, where you have depolarization of the ventricles following the atria, what if the ventricles don't depolarize until clear over here? Okay, that's going to be an indication that there's some kind of interruption in that pathway trying to get the signal to the ventricles. Okay, so that's that's good information. Okay, so that's something that's important to know. We might also see over time that someone's QRS complex gets higher. Okay, now what might cause that? Well, it might be caused by ventricular hypertrophy. What's that? That's when the heart muscle gets larger. Why might the heart muscle get larger? Because it's working harder, because there's some kind of failure in the heart system that means that the ventricle has to work harder. Okay, so that's another indicator. So you can get a lot of information about what goes on, what's going on in the heart just by looking at the electrical impulses. Okay, all right, so cardiac cycle. The cardiac cycle is basically, in the simplest terms, it's a repetitive contraction or systole and relaxation diastole of the heart chambers. Okay, so the left side pressure is a lot higher than right side because systemic circulation is more extensive than pulmonary. Okay, what does that mean? That means that your left ventricle has to squeeze out to your whole body. The right ventricle just has to go to the lungs. So the highest pressure is going to be in the left ventricle. Okay, so that's what we see. Blood moves through the circulatory system. Oh, I have that again, from higher pressure to lower pressure. Okay, so hopefully we know that. Let's see. Contraction of the heart produces pressure, right? Okay, so again, cardiac cycle, systole and diastole. It's two pumps that work together, left and right. We know that we have blood moving through. Wow, this is just a lot of repeated stuff. So systemic, pulmonary, and it's moving from higher to lower. Okay, I meant to delete that slide, I think, because it's repetitive. Okay, so let's go through the cardiac cycle. The cardiac cycle. And we're going to start with active ventricular filling. Okay, so here's a little clue. If we have something that says active, especially when I have it underlined, if we have active, that means there's probably passive. But since we're talking about a cycle, we have to start in a certain place. So probably right before this took place was passive ventricular filling. But we'll talk about, but that's going to end up at the end because this is, so if we're looking at a cycle that goes around, starts at one place and then gets back to that place, well, we're starting at this point, okay? And we'll end with that point, even though they're right next to each other. So, active ventricular filling, atrial systole. That means that the atria are contracting. Okay, the AV valves are open, and this is one thing you really have to pay attention to, is what's going on with the valves during each of these steps. A lot of it you can just sort of figure out. If the ventricles are filling, the AV valves have to be open. Okay, not only do the AV valves have to be open, if the ventricle is filling, you don't want it all just running out the other side through the semilunar valves. So the semilunar valves have to be closed. So if this is my ventricle, this is my semilunar valve that's leaving. This is my valve where it's coming in. Okay, that means this valve has to be open to let the blood flow in, but I don't want the blood to just flow right back out here, so that valve has to be closed. Okay, so this valve is open, and which is the AV valve, or if we're talking left side, we're talking mitral, right side, we're talking tricuspid, but that AV valve is open and the semilunar valve is closed. Because this is going on both sides. I tend to talk about systemic side because, it's bigger, more pressure, but it's happening on both sides. Okay, so depolarization of the SA node generates action potentials that spread over the atria, which is our P wave, and the atria contract. Okay, this completes ventricular filling. Also a hint, there was also already some blood in there. So the ventricles, the atria contract, squeeze the rest of the blood into the ventricle, and it completes that filling. So at rest, the contraction of the atria isn't even needed. Okay, that ventricle, all you have to do is kind of open the valve up from the body, and the blood flowing back from the veins. Oh, I shouldn't have made that blue, but the blood flowing back. I guess it doesn't matter, but it's coming back. It's going to just sort of slip right through those there and slip right into the ventricle, and it's going to start filling it, and then the atria will come through and contract. Okay, so during exercise, So at rest, you don't even need the atria to contract. It does anyway, of course it does, but it's not necessary. During exercise, atrial contraction is necessary because it increases the cardiac output like 3 to 400%. Okay, so 70% of filling happens passively, and this, what we're talking about now, is the active 30%. Okay, from the atria contracting. Okay, so now the ventricle has filled with blood. So here's our ventricle, here's our atria here. So we have this, and this is our valve coming through. So we have this period of time where the ventricle starts to contract. Now, remember in the last step how our semilunar valve was closed? Well, when the ventricle starts to contract, and it has all of this blood in it, okay, so it's filled with blood, it starts to contract, and that extra pressure is going to push that AV valve closed. Okay? This is kind of important. This isovolumetric contraction is way more important than people give it credit for. Poor little lost isovolumetric contraction. Now, so here we have this situation where the ventricle is contracting. It just pushed this AV valve closed, so that AV valve is closed, and the semilunar valve was already closed, so it could fill to begin with. And so now what's it doing? It's contracting, and it's pushing. It's not going to open that up. The papillary muscles are holding that, so it's going to generate as much pressure as it can to overcome the pressure, to overcome that closing right there. What's keeping that closed? The pressure in the atria, or in the aorta. So the pressure in the aorta is saying, nope, don't close, don't close, don't close. And then this ventricle will squeeze and squeeze until it overcomes that pressure and pushes that valve open. Okay, so now at that point we have, okay, so after this period of isovolumetric just means no change in volume. After you have that little period, the valve opens, the semilunar valve opens, and blood starts to flow in. Now, there's a lot to that, because you're wondering, well, that seems silly, but what's causing this pressure to build up out here? And that's exactly it. You could have blockages, atherosclerosis, arteriosclerosis, where you just don't have the holding capacity and the blood pressure is very, very high out here. If the blood pressure is high, then this guy is going to have to squeeze really, really hard, okay, to pop those valves open, or to pop the semilunar valves open. So if there's more pressure, this guy has to squeeze harder to get the valves open, to push the blood into the aorta, which means that you're going to have some hypertrophy here, maybe. And even if it does squeeze it out, it had to work so hard that it may not even get all the blood out before more blood is rushing back in, because the heart's not going to wait for it. So it's going to have to, it's not going to get as much blood out, or something like that. Okay, so contraction, both valves are closed, so no volume change begins at the completion of the QRS complex. Okay, well, I mean, the QRS complex is depolarization of the ventricles, which leads to ventricular contraction, so yeah, that makes sense. Ventricular muscles start to contract, increasing the pressure inside the ventricles, and initially those AV valves are going to close so it doesn't leak back into the atria. And then the semilunar valves that were closed, the semilunar valves were already closed, and so you've got squeezing. Okay, so isovolumetric just means that both valves are closed and it's squeezing. Okay, so 120 to 130 milliliters of blood are in the ventricles, and that's the end diastolic volume. Okay, so that's going to come back later when we start talking about preload and afterload. So the end diastolic volume is how much blood was in there as it filled up. It starts to squeeze, AV valves go closed, and there's about 120 to 30. That's the end diastolic volume normally. Okay, it's very brief. No blood is moving because all valves are closed. The goal is to overcome the pressure in the arteries. And yes, I know I was talking specifically about left side or aorta, but in the arteries, so these semilunar valves will push open. Okay, and then they finally open. Fantastic. Okay, the AV valves are still closed. Okay, so that hasn't changed. The AV valves are still open, but the semilunar valve popped open, and now blood is flowing out of the ventricle. Okay, so that's the second part of ventricular systole. Systole is contraction. Okay, so pressure in the ventricle is increased to the point where it's greater than the pressure in either the pulmonary trunk or the aorta. This pushes the cusps of the semilunar valves against the walls and opens the valve, and the blood starts to flow out of the ventricle. Okay, so blood is ejected from the ventricles. And then I mentioned earlier, so I'm not going to get too much into it, but the pressures are different from left and right. And as that left and right ventricles are different pressure. That's what that says. But after that first initial spurt of blood, so we're trying to push it out of the ventricle, then of course the pressure in the ventricle starts to drop, okay? Because it's moving that pressure out into the aorta or the pulmonary trunk, pulmonary artery. Okay, so at the end of ejection, 50 to 60% or 50 to 60 milliliters remain. So it's roughly half. Okay, so if we started out from 120 to 130, it's, roughly half of it's left. A little over half is left. Okay, has been ejected out. Now that's a reserve. So if you're exercising a whole lot, you might actually squeeze all of that out. Okay, during activity. Okay, so now we have a period of isovolumetric relaxation. So the ventricle has pushed its blood out, and then it starts to relax. Well, remember, the AV valves were closed to begin with, and so now the semilunar valves that just popped open are going to close again. The atria, or the aorta, I know I'm focusing on the left side, but the aorta has this pressure in it that it just got from having blood pumped into it, and now it's elastic, so it starts to squeeze. It's going to push those semilunar valves closed, hopefully, when everything is working right. Okay, so it pushes those semilunar valves closed. So there's a short period of time where the semilunar valves are closed and the AV valves are closed. Okay. But we can kind of predict what's going to happen next. Well, you've just lost pressure in the ventricles. So that means that maybe the pressure in the veins that are returning to it might be higher. Okay. All right, so completion of T wave results in ventricular repolarization and ventricular relaxation. Pressure falls very rapidly. The pulmonary trunk and aorta pressure is higher, and so that's going to close the semilunar valves. Okay, so that higher pressure is going to push back towards the ventricles, and those semilunar valves are going to close, and the AV valves were already closed. Okay, so it didn't. So when you were squeezing blood into the arteries, it didn't go back up into the whatever the oh gosh, I can't, atria. All right, so it wasn't pushing blood back up into the atria, so those valves were closed too. Okay. All right, so now we're almost done with the cycle. We have this thing called passive ventricular filling. So we just lost, okay, so if we pick up where we were, the ventricle has just squeezed out its blood. It only has a little bit of blood left in there. It's relaxed. Okay, the semilunar valve is closed, so no blood is flowing back in, which it's all out here in the, I know if I think left side, it's in the atria or in the pulmonary artery. Okay, so it's all out here. Here's our atria up here. Okay, and so we've lost this pressure, and now we've got blood flow returning to the atria, and now since we've lost all the pressure in our ventricle, well, now the blood returning to the heart is going to be comparatively a higher pressure. So it's going to push those AV valves open, and blood is just going to flow straight through. Think about this. It's going to flow straight through the atria and right into the ventricle. It pushes those valves open, and it's like, hello, ventricle, here we are, blood. Okay, and so that's your passive filling of the ventricle. The aorta, it's not really doing anything right now. It's just like this conduit, or this part of the venous tube that's of this return blood. So this return blood is coming back. It just kind of fills this ventricle because the ventricle is relaxed, low pressure, and so we have some filling. Okay, so that's this passive ventricular filling. And we know what comes next. Active ventricular filling when the atria contract. Okay, that's active. This is passive, so it's just sort of moving in. So atrial pressure rises above ventricular pressure and the AV valves open. Okay, so that's because of the return blood coming in, and blood flows into the relaxed ventricles, accounting for most of the ventricular filling. 70% is just this passive filling. Okay, so 70% of ventricular filling happens because blood passively flows in. No atrial contraction is required, but of course it still happens. And then we're back. We're back to the beginning. And this is another visual that kind of goes through the cardiac cycle all in one page. So you're welcome to look at that. Okay, so now if we kind of understand things that are going on with the cardiac cycle and with the heart beating, there are some other things, and I mentioned them before, and I said that, and one is preload and one is afterload. So let's look at here's a ventricle and here's the atria. Okay, so the atria, and there's the AV valve. Okay, and here is, if we say left side, we'll just say that this is the semilunar valve in the aorta. Okay, so as the heart, or as the ventricle, starts to fill with blood, it stretches. Okay? So anytime a muscle stretches, okay, remember you have the actin and the myosin heads that are sort of walking along. So I'll just draw this here. Myosin heads walking along. Okay? Now, when it stretches, these myosin heads start to kind of fall off the edge. Okay? And so you don't have, well, let me say this first, okay? Because I kind of went the wrong way. When it's very relaxed or very contracted. So here, let's look at this picture. This is a little better because this is what I'm trying to draw up here, and I'm doing a terrible job of it. Okay, so let's just focus on this. When you have the actin and the myosin that are trying to walk along, so the myosin heads are walking along the actin filament. When you stretch it out way too much, like you can see here, all of a sudden these myosin heads start to fall off of the actin filaments, okay? And you don't have a good contraction. When they're too close together, they get kind of bunched up, and they actually will start to fall off the other side. So you have, if I just draw a few, I'll try that way. If I have a few myosin, and they're all contacting the actin, that's a good strong contraction that can take place. Now, if I stretch it out or compress it too much, so let's say I stretch it out, and my actin is over here, that means these guys aren't doing anything. Okay, they're not doing anything. That's going to be a weaker contraction. Okay, so the same thing can happen if I compress it too much. Okay, so let's say that this is the direction of compression. If I compress it too much and these myosin heads, they can start falling off the other side. They'll bunch up with other myosin heads over here, and that's not a good contraction. So we have this sweet spot right in the middle. Okay, so that's what these pictures are kind of showing. Okay, so they're showing that you have good contact with the actin and the myosin, and you're going to have the strongest contraction that takes place in that area. When you get too much stretching, then they start to fall off. When you have not enough, then that's not good either. So what that means for us, and that's the point. Like, get to the point. When the more full the ventricle is, the better you are, the better your stroke volume is going to be. You're going to hit that sweet spot. And so as it stretches out, you're going to have more of a strong contraction. Okay, until it stretches so much that it actually is less of a strong contraction. Okay, so if we read this first line here, it's called the Frank-Starling law, first of all. In the heart muscle, more stretching means a more powerful contraction. So that's what this graph is showing. The more it's stretched, the more powerful the contraction, which is our stroke volume when it contracts. And so what this relates to, though, is volume, because that's what's stretching it. It's how much blood is in it. So the higher the preload, so we're preloading this ventricle. Remember, we're talking about the ventricle. So it gets loaded with blood, and it stretches. And the heart is like perfect. You know, a little bit of stretching is fantastic. It makes me stronger, and I can squeeze or contract even more strongly. So give me this volume, and I will increase my stroke volume. Okay, and I'll squeeze more blood out. I can handle it. A healthy heart, that's where it is. It says put a little more blood in, I'll squeeze a little more blood out. Okay, but in an unhealthy heart that is not able to get rid of all the blood or something like that, then the stroke volume is very, very high. Okay, so if you have high fluid volume, your stroke volume or your preload, the volume that's in there to begin with, is going to be very, very high. Okay, so preload is how much blood, yeah, just give us the definition. The preload is how much blood is in the ventricle before contraction takes place. Preload, okay? And that's volume. It's volume of blood in there before the contraction takes place. So in heart muscle, more stretching means more powerful contraction. More blood, ventricular preload, is the amount of stretch caused by the blood entering the ventricles. Okay, so it's based on volume, the volume of blood entering the ventricles. And again, the more blood, the more forceful the contraction up to a point, because it all falls apart here, and this is heart failure. Okay, it's a form of heart failure. Okay, so if we have preload, you know we have to have afterload. So afterload is pressure. Now all that blood that was in that ventricle has to be squeezed out. We're going to say through the aorta, okay? Because we're talking about, it's ventricles, so it could be left side or right side. We're going to say on the left side. Now remember earlier when we had this isovolumetric contraction where the valve was closed, and we said, oh, you got to generate enough pressure to push that valve open. That's kind of related to afterload. So that means this ventricle has to squeeze, and it has to push these AV valves open and out into here. Well, what if out in here we have clogged arteries? We have atherosclerosis. You know, we have a mess out here. Okay, I'm kind of maybe exaggerating, maybe not. But we have this mess out here that doesn't let the blood flow nicely. And so here's this poor ventricle, doesn't know what's going on. It's squeezing, it's saying, I'm doing the best I can. I'm trying to push the blood out into the atria or into the aorta, but there's this blockage here, and so it has to generate a whole lot more preload. Or afterload, I'm sorry. It has to create a whole lot more pressure. So this afterload is the pressure, and usually that's back pressure from the aorta. Okay, so afterload is pressure that's caused by resistance of the aorta or the pulmonary artery. I don't mean to leave that one out. The aorta or the pulmonary artery to blood that's flowing into it. So usually it's caused by some kind of stenosis, or atherosclerosis is very common, some kind of higher blood pressure situation out here. Okay, so it's caused by aortic valve. Okay, but I didn't mention that. Yeah, what if your valve, your aortic valve wasn't working right? Okay, so that could cause an increase in pressure. So it's trying to push the valve open. If the valve isn't functioning right, that could cause problems too. So it's caused by aortic valve issues, stenosis, narrowing, and high blood pressure. Okay, so this afterload pressure can be related by anything afterwards saying, nope, we don't want the blood, or the blood can't come in, or somehow you're preventing blood from flowing in. Okay, so the more resistance, the less blood is released. Think about that. If here's my ventricle that's trying to push the blood out, it's got its job to do, it's trying to do its job, it tries to push the blood out, there's resistance, there's an increased afterload, and so it's not going to get the blood pushed out. So that's kind of what I mentioned earlier when I was going through the cardiac cycle. And so that's another source of failure. That means you're not going to get the cardiac output. Your cardiac output's going to drop, and those poor cells that are waiting for the blood are just going to have to wait. Okay, they'll get some, but it's not enough, and so that's a heart that's having problems. Okay, or heart or a circulatory system that's having problems. Okay, different drugs affect preload or different afterload differently. Remember, preload is volume, so if you reduce blood volume, you're going to affect preload. Afterload is caused by resistance, so if you do something that causes like vasodilation or vasoconstriction, that's going to affect afterload, okay? So keep those straight if you can. All right. Okay, so now I've thrown around a couple of terms. I've already been using it. Stroke volume. Stroke volume, here's our ventricle. Okay, here's our end diastolic volume. So that's where we had passive and active ventricular filling. Remember those from the cardiac cycle? This is our volume, okay, before contraction. And then we have ventricular contraction, and this is our volume after contraction. We've squeezed out a whole bunch of blood. Good job, okay? If this is so we call this end diastolic, so this is after it was relaxing and taking in the blood and saying, yes, I am the ventricle, I will squeeze the blood, you give me the blood, here's all the blood. The end diastolic volume is how much blood was in there to begin with. Then it squeezes, and that's systole, it squeezes all that blood out, and that's all it's got left. Okay, so after systole, that's how much blood is left. So what's our volume of that stroke, of that contraction? What's the volume that left? At least this actually looks like a wine glass. I wonder what that means. It's going to be the end diastolic volume, which is what I was trying to draw here, and then the end systolic volume, which is here. So the blood that flowed out is our stroke volume. Okay? So the calculation of stroke volume is end diastolic volume minus end systolic volume, and that's, so basically the simplest way, stroke volume is how much blood your ventricle just squeezed out. That's what stroke volume is. So that's why it's an easy term to use, but then when we try to define it, we start using words like EDV, ESV, end diastolic volume, and systolic volume. It's related to that very, very directly. That's absolutely what it is. Okay, so at rest, every beat of the heart pumps about 60% of the blood. I was saying earlier, a little over half, so about 60% of the blood, or 70 milliliters. And of course, that can change with demand. You can increase stroke volume. If you're exercising, you start running, something like that, doing jumping jacks, you will increase your stroke volume because you need to squeeze more of this blood out. So maybe instead of 60%, you squeeze out 90%. Certainly possible. You can squeeze out 100% of the blood. Okay. All right. So this is important to preload. Higher volume results usually in a healthy heart in a larger stroke volume. Okay. So if you have more blood that moves in, if the heart is healthy, it squeezes just right, and it's going to squeeze a larger amount of blood out. Afterload, if there's too much resistance, it's going to prevent that ventricle from pushing the semilunar valves open, and then your stroke volume will be reduced. So too much resistance or a higher afterload is going to decrease your stroke volume. Okay. Contractility, how strong is my heart? How strong is that heart muscle? Was it stretched out so much that it wasn't very strong? I mean, because that can happen if your preload is too high, it stretches so much. But however that heart, that ventricle is contracting, did you have a heart attack? Are myocardial cells or muscle cells dying because they didn't get enough, because you had a heart attack? That's going to be weaker, which is going to reduce your stroke volume. Okay? Your stroke volume means everything to those little cells out there. Those little cells that are waiting for blood, they depend very strongly on a stroke volume that matches their need. Okay, so if your heart is not working right, these guys are going to suffer. Okay, unless you have a compensatory mechanism like an increased heart rate. Increase the heart rate, sure, you can deal with a smaller stroke volume, but you're going to have to. But that's going to. That segues nicely into something called cardiac output. Okay, cardiac output is when you take your stroke volume and you multiply it times your heart rate. Okay, so that's the cardiac output. So whereas stroke volume was just saying how much blood is leaving the ventricle, so this is a ventricle. Now all of my ventricles look like wine glasses. So how much blood is leaving, that's your stroke volume. Your cardiac output is going to be over 1 minute. So cardiac output is saying, okay, sure, your stroke volume is down, but is your heart rate compensating for it? Okay. Because if your heart rate is compensating for it, then the little cells out here that are living out here are all happier. Okay, because they're, as far as they're concerned, they're getting all the blood they need. Your poor heart, though. And so cardiac output is heart rate times stroke volume. We just talked about what stroke volume is. I hope I don't have to explain heart rate. Okay, so normal cardiac output is about 5.25 liters per minute, so that's normal, and that's really 70 milliliters. Actually, that doesn't need to be 70 milliliters per minute. That's just 70 milliliters times 75 beats per minute. That's how we got that. We got that number. So in a healthy system, stroke volume is fairly constant. If blood volume drops or if the heart weakens, then stroke volume declines. Stroke volume declines, and cardiac output is maintained by increasing heart rate. Okay. So this kind of looks at everything and talks about how all of these things are related: preload, afterload, heart rate, contractility. We've mentioned all of these things, so you can kind of look through this on your own and try to piece together. Okay, I see how these things are related to each other. Okay, because this is already a really long lecture, and I'm getting a little loopy. Okay, so I put this on here also so you can kind of see all of the things that we're measuring at a glance. Okay, so EKG is measuring electrical. Remember that, electrical. It's not necessarily measuring pressure. It's not at all measuring pressure. It's, as far as the EKG goes, the ventricle didn't even have to have contracted afterwards. All it's doing is saying it depolarized. Now the contraction is going to follow the depolarization. So if we look at the pressure here in the ventricle, then yeah, it's going to depolarize right after, or it's going to contract right after depolarization, but it's going to right, so it follows the electrical event. So then there's the aorta. The aorta is going to increase in pressure as the ventricle, because this is pointing to the dotted line. I'm going to make this the ventricular pressure a little bit higher or darker. So as the ventricle contracts, the aorta is also going to increase in pressure. Okay, so it's going to follow nicely, but it's not going to drop all the way because these semilunar valves snap closed and saying, oh, you wanted your pressure to go to 0, did you? Like the ventricle, but it can't. Okay, you're going to have to keep that pressure to keep those blood vessels open. Okay, so pressure in the aorta maintains an elevated diastolic pressure close to the BP we measure. Okay, so that's actually what we're measuring. When we measure blood pressure, we're actually measuring ventricular systole. Okay, specifically left ventricular systole. That's what we're measuring. Diastolic pressure in the ventricle drops to almost 0. Okay, that's because it doesn't have that semilunar valve. It actually opens itself up to lots of different things. Okay, so here we can see the heart sounds. So the first sound is when the AV valves close. That would be your mitral and your tricuspid. When those valves close, it sort of goes, and you have that turbulent flow for just a little brief period of time, and you can hear that. Then the semilunar valves close, and that's going to cause the same sound, and that's going to be your second heart sound. Okay, and then you can see some other sound of turbulence, like in the aorta, or filling turbulence, and those can be like when the ventricle is filling, you might be able to hear that sound too. Okay, but normally we just look at the lub and the dub. Okay, and so that kind of puts all of that together. Okay, so you can kind of look at that and see which things and all of it should make sense to you. Okay, so you should be able to say, okay, yeah, that makes sense, that makes sense, that makes sense, as you move down. Okay, veins return blood to the heart. We've already talked a little bit about veins. They're very compliant. Okay, so compliance. Compliance just means that you do what you're supposed to do. And if we're telling a vein, hey, stretch, then they are compliant and they stretch. So in the case of cardiovascular, when we say that a vessel is compliant, that means it is able to stretch. Now, we're not talking about elasticity. We're not talking about its ability to return to its original size. We're just saying how easy it is for you to stretch. Okay? Are you very compliant? We could look at like a plastic bag. at like a grocery bag or something like that, and here's this grocery bag that you have it all wadded up, and you blow into it, yeah, it's gonna go out full size right away. It's very compliant, but it's not very elastic. Okay, so yeah, when it's small, it'll take on lots of air very, very quickly, very, very easily. Same thing with the veins. They're very compliant, but they're not quite as elastic as they are elastic, but they're not quite as elastic as the arteries. Most 60 to 70% of your blood is in the veins. Okay, so that's all of this area here. Okay, all of this veins. Okay, and then the rest is kind of distributed to everywhere else. Okay, so here you have some blood in the lungs. Um, there's your heart, and then the arteries, systemic arteries, lung arteries, um, and then the capillaries. Very little volume, but there are tons of them. Okay, veins. So I'm going to go through this pretty quickly because we're really long here. Valves are found in all veins greater than 2 millimeters in diameter. Okay, so the purpose of valves is we've got this loss of blood pressure. The blood pressure is decreasing as it goes through the veins. So one way we can keep blood flowing is to push them through veins, and these veins, push them through the veins through the valves. And then these valves are one-way valves. So that means when it's pushing in this direction, they open, and then they very quickly close, and they will keep blood flowing backwards toward the heart. Okay, so two flaps that overlap. There are more valves in veins of the lower extremities than in veins of the upper extremities. Why? For the obvious reasons, because it's harder to push against gravity here than it is like against gravity here. Okay, because this is where your heart is. It's trying to get it back to the heart. Okay, so you have all of these veins that are pushing, or the veins with the valves. Your muscles are squeezing those veins. Okay, so here's your little vein in here, and you've got a muscle that's going to push on it. It's going to push the valve open. Blood is going to flow up, and then it's going to, so moving your muscles, contracting your muscles is what causes those squeezes those veins and causes those valves to open. Okay, one other thing, I'm not going to say too much about this, but lymph vessels return blood to the veins. We've already talked a lot about lymph vessels, so that's all I'm going to say. That as things move into the lymph vessels, that lymph fluid, the fluid that moves into the lymph, will eventually make its way back into the veins after it goes through the spleen and all those lymph vessels. Okay. Oh wow. Okay, that's it. I thought we were going to talk about something else, but yeah, that's the end of this section.